It's kind of hard to have pictures as pretty as like the whole cage is coming together, so mine won't be quite as pretty as Cyrus's, but hopefully these are a lot of like real world examples of things that I've kind of gone through, and hopefully it will maybe streamline some of your downstream stuff. So as Cyrus was saying, there are a lot of examples of symmetry occurring in nature. If we go not quite as big as a virus, but we go smaller, like hemoglobin has something called C2 symmetry. Porins inside of your membranes, which allow trafficking, have C3, and limousine synthase would be something considered C5. But if we do go up in size to something like a virus, we can have something called icosahedral symmetry, which has a mixture of this like C2, C3, and C5 symmetries present all on the surface. And this kind of poses an interesting question from like an immunology standpoint, or if you are working with like a protein that you want to do something entirely different with. But can we really stabilize like these individual symmetric axes using diffusion? And so like with these, if you express them solubly, they will all come out as monomeric proteins. But using Rosetta fold diffusion, we are able to generate specific scaffolds that are de novo designed to stabilize like these specific structures. There are a few important considerations when you do want to use diffusion in a symmetrical manner. And as Amrita mentioned earlier, the z-axis alignment is actually really important. Classically in Rosetta, whenever you're handling diffusion, it was always around the z-axis, and that same trend is carried forward into Rosetta fold diffusion as well. So if you input a structure into diffusion without aligning to symmetry, you end up, diffusion kind of assumes that you did it properly, and it will put the z-axis through your structure and then twist it into some like mess, which you can see on the right up here. But the structure on the left is actually what I thought I was putting into it. And then when we do align it to the z-axis, it is maintained properly and carries forward. If you're not doing symmetrical diffusion, this isn't important. Like if you put in just for a mini binder, you can generate whatever you want without respect to axes, and you'll be totally fine. And in the activity that you guys just started right now, where you basically just told it to generate a C4 symmetrical thing without any input PDBs to kind of model around, you also don't need to worry about symmetrical axes. It only matters if you want to specifically scaffold an existing thing. And if you just press forward with a misaligned structure, so this is the structure I put in, and this is what diffusion returned to me. So yeah, it really does not work. The script to align to the z-axis actually took about 2 days to kind of get all of the parts to work. It is available on the GitHub for this like workshop. So if there's ever a chance you might need it, I highly recommend grabbing it because it'll make your life a bit easier downstream. Another thing to consider for diffusion is your input size. So Cyrus kind of mentioned this before, where you really do want to kind of like minimize the thing to reduce the compute time and hopefully get better structures. One example of this is I put in the structure on the left. There was over 2,700 residues, and diffusion output the thing on the right. It was right at the edge of what was able to be held in the memory of the GPU, and it really just kind of like destroyed everything. So to get around this, you are able to modify your input structures, and you want to do this in a way where you kind of consider what's important. So for me, like generating something around a C5 axis of symmetry, I could kind of just draw a red circle and then cut away everything else. And that left just this structure right here. And what I effectively did was threw out 1,600 residues, which were not necessary for like what the model had to consider to generate a like usable input. So now I put that on top of this, and we were able to get like a nice formed structure. So yeah, and then we were able to get sequences, and this like works. So again, like with your input size, you do want to trim. With your input size, you do want to trim, but you also want to consider your downstream goals and downstream abilities. So for example, a common filtering step, or like the next step for a lot of labs these days, is using something like an MPNN to AlphaFold script to figure out like if this is possible to fold decently. But if you run AlphaFold on a massive structure, it very commonly kind of makes nonsense and also takes a really long time. So for like this specific problem where I was just trying to see if the central component was like feasible, I actually was able to use a script which removed the entire like top and the entire bottom piece and only run MPNN AlphaFold on this 3-helix bundle. So to trim the sequence, we basically wrote a script which just looked for a consecutive string of more than 5 glycines, and then it would just copy that string of more than 5 glycines into a new PDB. It was kind of the easiest way because with most of these structures we were generating, we were allowing diffusion to pick somewhere between like 80 and 100 residues rather than being like always 85. And just by removing like the polyglycine backbone that diffusion outputs, we were able to like very quickly just run on an entire directory and move that into a new one. Another thing, so going back to the first goal, one thing I mentioned was like I wanted to stabilize the like pentameric or the C5 axis of symmetry. But if we look here, like if this was a stable interface, this would definitely act as like a solid connection point to the bottom piece. But that wasn't a stable interface, so while I was able to tie together 5 proteins well, it didn't hold them in like the naturally existing conformation. So the next step that I moved to is diffusing in steps. And what I mean by this is basically like diffuse with a specific goal in mind and then use that diffusion output to carry forward into your next goal. So in an attempt to stabilize the C5 like interface, I used that same input that I had at the beginning, and I realized that in red you can see the diffusion structure, but I trimmed too much. So there's that fine balance of like leaving just enough of your starting structure where you aren't creating something that would have clashes with like the naturally occurring backbone, but not so much that it like really slows down the model time or makes things just like impossible to actually run. And so to get around this, I actually put back like this extra piece of the protein. And this could be really important because like let's say one of the activities later will be putting like an influenza component on top of that I5350 nanoparticle from the King lab. And as a test run, I basically deleted the majority of the King lab protein, but diffusion will fill in the holes because diffusion is trained on like real protein structures. So having like odd holes, it doesn't like and will try to fill those. So you want to leave enough of the structure that it behaves as you're intending, but not so much that it either can't run or it takes hours to generate a single structure. But once we put back the additional residues, we were able to generate this scaffold, which packed pretty nicely against the bottom of the protein. I only made one, actually, expressed it, and it did form a pentamer. So this definitely is like a very viable way of trying something. I might test more than one in the future. But once we had this bottom model, I then put that back over the top of the original thing that I was considering scaffolding to. And now I was able to generate this connection piece, which, like, this looks just like a much more stable protein. The bottom protein expresses around 20 mgs per liter, and with the connection piece, the best expressing one was over 30 mgs per liter, so I actually got an expression bonus from the existence of the connection, and I would say all of them fell somewhere between 10 and 30, so there wasn't too much of a hit, even in the worst cases. Okay, so while it is important to trim, you want to make sure you don't trim too much, because diffusion will find those holes and then try to work backwards to like fill what you cut away, even though it's not really gone. And then diffusing in steps allows you to form a good connection, and then from there kind of go on to the next step. So like I was trying to make a mini binder against a specific target, but diffusion was having a really hard time placing like any of the structures in a reasonable way against it, because the target kind of was convex and it had to like bind against that surface. But I was able to use in steps diffuse in like one helix, which was in like one specific place, and then use that as the starting point for the next helix to go into like the next place that I was hoping to get. So you don't have to do this for like just big things. It can help you solve problems on small things as well. And then finally, again, in an effort to reduce computational time, you absolutely do want to figure out a way to filter your outputs from diffusion. So yesterday, Amrita was showing that like we can use the radius of gyration on small proteins. For larger proteins, typically secondary structure filtering is very effective. So here's an example of 17 outputs from diffusion. I don't know if you can see super well, but a lot of them end up being kind of just completely unstructured proteins. A lot of them end up also just being like these beta barrels, which likely wouldn't be super soluble once expressed in solution. So using a secondary structure script, we were able to remove away pretty much 90% of the outputs, and that left us, with that one in the bottom left, which ended up becoming the one that we expressed. So secondary structure filtering cut down like a whole directory to these 6, and then I ran this all through MPNN AlphaFold, and that one was the only one that scored well. MPNN actually fixed part of the unstructured region and led to the final structure. But to generate this one structure, I had to generate roughly 400 diffusion backbones for like one single structure to come out of it. So it wasn't a very good like pipeline to have to generate 400 for one single like positive in silico result. So I used this as a starting structure to kind of seed for partial diffusion. And I think you ran some partial diffusion yesterday, and there were some questions about like how many time steps to actually get diversity. So what I did was I passed in this whole structure. I told it to hold the N and C termini constant, and then diffuse in the middle, like partially diffuse the middle. If you do the 10 time step partial diffusion that was like built into the program, what you see is that it really acts kind of like a backbone rub. You might have some local rearrangements. It might kind of make the secondary structure look a bit nicer, but it doesn't drastically change like your outputs. So with 25 time steps, I was able to get variances of 3 helical bundles, but they do actually have some change now. And when I went to 35 time steps, we started to have some like pretty significant backbone rearrangement. So all of these are actually like sequence optimized, and they'll be tested next week. So I can't tell you like how it worked, but one other thing, kind of going back to what Cyrus was talking about as well, is unfortunately, if you like put this pentameric scaffold underneath like my pentameric protein, AlphaFold really struggles to predict that sequence properly, or like predict that structure properly. So what you can do is run like protein MPNN or any of these new machine learning models, and then use Rosetta docking and packing to like pack those side chains and kind of visualize the interface that you are having. So again, it kind of goes back to like the limitations of the software that we have today, and then being creative about like your ways forward. So when you do the secondary structure filtering, especially on the bottom or like as a scaffold protein, typically I find that 30 to 35% cutoff ends up being pretty like strict enough on the coils and turns. So coils and turns are what lead to that spaghetti-like structure, and below 40% on beta sheets, just so you don't have a full beta barrel. It is nice to have some, just for like some structural diversity, but you don't want an entire beta structure. And then for partial diffusion, you can hold some areas of your structure constant and then noise an area of interest. So if you want it to maybe move a little bit closer to your like target, if you're trying to make a binder, or if you just want to generate different types of scaffolds to go underneath and stabilize a specific interface, those are all possible. So you can kind of be creative. And then finally, highly recommend iteratively optimizing your sequences. So on the left was the original MPNN prediction of the MPNN sequence predicted by AlphaFold of the structure that kind of became our final design. And on the right, it went through iterative optimization through a pipeline that Amrita made called EvoPro. And you can definitely tell that it's a bit more confident with the new structure. And this works on proteins of all sizes. So this was one of like the larger scaffolds that I made. On the left, I had MPNN predict the middle structure, and then AlphaFold 3 fold the whole thing. And you can tell that it wasn't the most confident. And then through like iterative steps of sequence optimization, so you basically like take this structure out, take this structure, put it back into protein MPNN, have it repredict the sequence based on like the minor changes that happen when it predicts the new structure. And then unfortunately, I had to like manually by hand redesign everything, which I'll talk about in a second. But you were able to get a much more confident structure. And then through progressive rounds, I got the final design on the right, and that was the one that actually increased the expression of the final construct by 30 or 50%. And then to go about this manual redesign at this point, the best thing that I've found, and this can also work if you're trying to like reduce hydrophobic patches within specific areas of a protein, because you are having solubility issues, is I run protein MPNN probably 500 to 1,000 times, and then I look for maybe the less prevalent mutations or the like potentially new interactions that could be formed. So a lot of these maybe like we'll just repredict the starting one, but at the end over here, protein MPNN really loved putting alanines on this final helix, which is what connecting to the base. but that was going to be very solvent exposed on all sides. So I really did not want to do that. So this might be really small / hard to see, but like protein MPN really wanted an alanine at this position, but you can see that like there was a slight light blue of glutamate there. So rather than taking the protein MPN predicted sequence just like as the most prevalent one, I looked for less common mutations at those positions that were more like sensical in the context that I needed them to be. Another thing you can try doing is reducing weights or like penalizing specific amino acids, or I didn't want to introduce cysteines at all. So I just told it like it was not allowed to put a cysteine. And then that is another way to kind of slightly bias a sequence away from something that could be disadvantageous for your specific needs. But yeah, and then at the end, when I would like generate a new sequence based on like this map, you can score it using either AlphaFold or Rosetta to like repack everything. This is soluble MPN. Yeah. So the question was, what is the reason protein MPN put the alanine at that surface exposed region? We have no idea. This is something that I've seen in a variety of designs in the past, and we've ordered, like just for testing so that we better understand, because maybe there was something that like we just missed. I ordered like the alanine patch, and then I ordered the redesigned like charged patch, and the redesigned charged patch expressed a lot better and acted as a better, it like achieved the goal of that protein better than the alanine patch was. So I don't know why sometimes protein MPNN does this, but I have found it to not always be great. So you definitely could output the probabilities. I guess the reason I didn't output the probabilities and went with this method instead was because it was easier for me to visualize, like if there was a glutamate that was predicted at this position and then like an arginine at this position, or like if there were less probable outputs, it was still easier for me to find those rather than just looking at the probabilities. But yeah, you absolutely could use the probabilities. It just wasn't quite as easy to look at like a whole structure in that method.